Hi guys, welcome to this WACE Academy online lesson on ethanol. In this lesson, we will be covering dot points 5 and 6 of the subtopic Chemical Synthesis of Unit 4. To begin this lesson, we'll first look at the role of enzymes in fermentation and how this is used to produce ethanol. Then we'll learn about the hydration of ethene as an additional process used to produce ethanol. And finally, the production of the ester ethyl ethanoate. An enzyme is an organic biochemical catalyst, most commonly a large complex protein that increases the rate of biological chemical reactions by lowering the activation energy. They are not used up in the reaction, but may be destroyed if they are not kept within their optimal condition ranges. Those of you who do biology may be familiar with such things. Therefore, using enzymes in industrial reactions reduces the required temperature and pressure, and thereby increasing the economic viability, or cost effectiveness. The best way to understand the advantages of using a catalyst is to look at an example. So let's use the production of ethanol. Ethanol can be produced by hydrating ethene or from glucose through an organic process called fermentation. We're going to cover both of these methods, but we'll begin with fermentation. So during fermentation, sugar or glucose from plant material is converted into ethanol and carbon dioxide in the presence of a yeast catalyst. This usually takes place at temperatures of around 37 degrees Celsius and 1 atmosphere of pressure. The balanced chemical equation for fermentation is glucose, which is C6H12O6, is converted into 2 ethanol molecules and 2 carbon dioxide molecules. This reaction requires an enzyme, so it's essential you include it just above the arrow. Yeast is the microorganism that provides the enzymes for this reaction, so you can just write the word yeast. The necessary reaction conditions for fermentation are 37 degrees Celsius for optimal function. A good way to remember this is think about the temperature your body operates at. All the reagents must be dissolved in an aqueous solution, again like in your body. The reaction must be performed in the absence of oxygen, which we call an anaerobic environment. We also must have the yeast enzymes. So the drawbacks for fermentation to produce ethanol is that there's a slow rate of reaction and is thus time consuming, and stops when ethanol concentration reaches around 15%, but this often varies by a few percent depending upon the type of yeast you use. It must also be performed in batches instead of continuous production. To purify the ethanol, a process called fractional distillation is used. However, another positive side of fermentation that we haven't looked at yet is the fact that it's renewable, as it uses enzymes and reagents that can be grown, aka sugarcane. Now that we've completely covered fermentation, let's move on to the second method of producing ethanol. So this is what an ethene molecule looks like. And this is what ethanol looks like. Notice that they are quite similar. Both of them have that 2-carbon backbone surrounded by hydrogen atoms, with the only difference being that ethanol has an extra 2 hydrogen and 1 oxygen atom, as well as having a single bond between its carbons, not a double, like ethene. You might have also noticed that the difference between these two hydrogens and oxygen, is also the composition of water, and we're going to use this to our advantage to convert ethene to ethanol. Specifically, ethene, when reacted with a water molecule, produces ethanol, like so. When we compare the reaction conditions to fermentations, we notice a massive difference. This reaction must be carried out at around 300 degrees Celsius and under 60 to 70 atmospheres of pressure. And instead of using an organic catalyst like fermentation does, this reaction uses phosphoric acid as a catalyst, coated on top of silica. The advantages of using this reaction over fermentation is that it produces a much higher percentage yield, almost 100%, therefore producing a much pure final ethanol. The reaction can also be carried out continuously. However, the hydration of ethene isn't as simple as we first thought. In fact, there are a few steps you must understand. Now, it may take rewatching this section a couple of times to grasp it, but once you understand it, it's a fairly straightforward process. So, within the reaction vessel, we have an ethene molecule, a phosphoric acid molecule as our catalyst, and a water molecule, which at 300 degrees is steam. The first step involves the removal of a hydrogen atom from the phosphoric acid, and this temporarily bonds to a carbon atom on the ethene, which causes the double bond to break apart. Once this happens, the new ethene molecule will look something little like this, with the red hydrogen being the one that has moved over from the phosphoric acid. The next step involves our water molecule. So notice how the other carbon has only 3 bonds attached to it, and we know that carbons must have 4. So this leaves this current rearrangement a little unstable, and so the water molecule will come and bond to this remaining carbon. And so as the water molecule comes to bond with the ethene molecule, we now have the first red carbon from the phosphoric acid and our water molecule attached to the other carbon. So at this current organisation, the two carbons are happy because they both have 4 bonds. However, the oxygen has 3, one to the carbon and its 2 hydrogens, and we know that oxygen can only have 2. And so this hydrogen from the water molecule moves over and attaches to the first carbon, and in doing so replaces the original hydrogen from the phosphoric acid, that then moves back to the phosphoric acid molecule it first came from. In doing this, the phosphoric acid molecule is essentially regenerated because we know that in order for it to be a catalyst, it cannot be used up in the reaction. And so because it's regenerated, it returns to its original full form and is able to be used in the next lot of reactions to produce more ethanol molecules. And so now we have a complete ethanol molecule and a complete phosphoric acid molecule. The summary of these steps are: 1. A hydrogen ion breaks off from the phosphoric acid molecule. 2. This joins and then breaks open the double bond on ethene. 3. The water molecule attaches to the remaining carbon on ethene. 4. The original hydrogen from the phosphoric acid breaks off and rejoins the phosphoric acid, and thus regenerates the catalyst. 5. The hydrogen ions rearrange to form a complete ethanol molecule. Now we've covered both fermentation and the hydration of ethene, let's compare them both side by side. So fermentation is catalyzed by an organic catalyst called an enzyme, in this case yeast, and as such is able to be carried out at 37 degrees Celsius and 1 atmosphere of pressure. The reaction produces only around 15% of ethanol. It is considered a renewable method of production, and all reagents must be dissolved in water and carried out in the presence of no oxygen, which we call anaerobic conditions. Hydration of ethene, on the other hand, is catalyzed by phosphoric acid and is performed at 300 degrees and 60 to 70 atmospheres of pressure. It also produces a near 100% concentrated ethanol, but requires non-renewable reagents, ethene, which we get from crude oil. Now we have these methods of producing ethanol down pat, let's take a look at some further reactions we can perform with ethanol to produce some new chemicals. So one chemical we can produce from ethanol is ethyl ethanoate. This reaction is called esterification, which in general produces chemicals called esters, with one such type being ethyl ethanoate. Esterification reactions begin with a carboxylic acid and an alcohol, and form an ester and water. They also require a concentrated sulfuric acid catalyst. Naturally occurring esters are found in fruits, and they provide the flavour and odours. Ethanoic acid reacts with ethanol in the presence of concentrated sulfuric acid as a catalyst to produce the ester, ethyl ethanoate, and water. However, this reaction is slow and reversible, which means that some of the product can be converted back into its reagents. To reduce the chances of this reverse reaction occurring, the ester is distilled off as soon as it is formed, which lowers its concentration in the reaction vessel, and thus pushing the equilibrium of the reaction forward, causing it to produce more ethyl ethanoate. The concentrated sulfuric acid removes water from the products, which catalyzes the reaction. We can consider the production of esters, such as ethyl ethanoate, to have multiple stages in their chemical synthesis pathway. We first began with ethene from crude oil or glucose from sugarcane, which was then formed into ethanol. From here, ethanol undergoes a reaction called esterification to produce the final ethyl ethanoate. Now that we've nailed the reaction sequences, we've come to the end of the lesson. I hope you enjoyed it, and I'll see you next time.